Multi-Omics combines several layers of biological data to give researchers a more complete view of disease, treatment response, and patient differences. These layers can include genomics, which examines genetic variation, transcriptomics, which looks at gene expression, proteomics, which studies proteins, metabolomics, which measures small molecules linked to metabolism, and epigenomics, which examines changes affecting gene regulation. Looking at one layer alone may only explain part of what is happening biologically. A genetic variant might suggest a particular risk profile, for example, without showing whether the relevant pathway is active or how proteins and metabolism are being affected. Analyzing several layers together can help researchers connect these signals. This is particularly relevant to personalized medicine, where patient characteristics are used to inform prevention, diagnosis, or treatment. In clinical research, multi-omics may help identify likely responders, patients at greater risk of progression, or groups that may require closer safety monitoring. Oncology provides a familiar example. Tumors can be characterized by their molecular profile as well as their location. Combining genomic, proteomic, or metabolomic information may help researchers identify biological features associated with treatment response or particular patient subgroups. A study might collect biological samples before treatment and again afterwards alongside standard clinical assessments. Researchers can then explore molecular signatures associated with response, safety findings, or disease progression. These analyses may support exploratory biomarker work, patient stratification, endpoint interpretation, or the design of later studies. More data, however, does not automatically produce better evidence. Multi-omics datasets can contain thousands or even millions of variables. Separating meaningful signals from noise requires a clear clinical question, careful data processing, and appropriate statistical planning. Integration can also be difficult. Different omics layers may come from separate laboratories, platforms, sample types, or processing pipelines. Statistical approaches can include Bayesian integration, network analysis, dimensionality reduction, and machine learning, alongside models that link molecular data with clinical outcomes. The method should follow the research question and the level of evidence required. Machine learning can help identify complex patterns or combinations of markers, but models can overfit the dataset used to develop them. A promising result needs to be reproducible, interpretable, and biologically plausible before it can support stronger conclusions. Operational details also affect whether multi-omics data can be interpreted reliably. Sample timing must align with treatment and visit schedules. Subject identifiers, visit labels, dates, and time points need to connect laboratory outputs with the clinical database. Missed samples, dosing interruptions, or samples collected outside the planned window may also influence the analysis. Single-cell and spatial approaches add further detail by examining individual cells or the location of molecular signals within tissue. They can help explore areas such as tumor heterogeneity while increasing the importance of consistent sample handling, processing, and documentation. The starting point for any multi-omics study should be the clinical question. The study, design, and data flow should then be built around it. Molecular signatures may be scientifically promising, but they need suitable validation before they can support patient classification or treatment decisions. To learn how Quanticate can support your next trial, email inquiries@quanticate.com or request a consultation through our website. Thanks for watching, and don't forget to subscribe for more practical insights on clinical trials and modern drug development.